All right. Hi, everybody here today. Miss Fennell with you to do the Catalase Lab so you can see what happens in the Catalase Lab. So for those of you who attended class today, I'm going to do the notes piece of this first, and then I will do the lab. So if you were already in class today and you already heard these notes at the beginning, you can just fast forward to the part of the lab where I actually start working with the stuff. It's probably about 2/3 of the way through. The 12-minute video here or so is me actually doing the lab. So let's go over the background that you need to know to understand what happens in the lab. We've been studying DNA. This whole chapter is about DNA, and DNA is instructions for making proteins. That's how it controls everything in the cell. So how does DNA control a cell? By giving directions for making proteins. What do these proteins do? Well, they control all the chemical reactions that happen inside the cell. So they're going to build cell parts and control chemical reactions, and that's how the DNA is able to control everything that happens inside the cell. Okay, so there are literally thousands of these genes that make thousands and thousands, probably 100,000 proteins inside the cell. So we're going to work with one example of one protein that is found inside our cells today. It's called catalase. So there's a gene that a lot of living things have, most living things have, that will make this protein called catalase inside their cells. So catalase is a protein that controls the breakdown of a waste product. So as cells are doing their thing, doing all these chemical reactions, they produce waste products. One of the waste products is actually a chemical you might be familiar with outside of cells. It's called hydrogen peroxide. So the job of this protein catalase is to break down this waste product that builds up inside of cells. If too much of this hydrogen peroxide is left to build up in the cell, it will poison the cell and the cell will die. So we actually have a gene that makes the protein catalase that breaks down this poison inside of our cells, if that makes sense. So hydrogen peroxide is kind of interesting. It is a waste product that builds up inside of our cells, but we can also buy it at the grocery store, right? Which is kind of weird and different. So the chemical formula for hydrogen peroxide is H2O2. Just one more oxygen than water has. What's weird about this chemical, though, if you just let it sit around, if you just put it in a glass, put it in the dark, and you let it sit around for a month, it will naturally break down into water and free oxygen. It will break down into H2O and O2. It will just naturally do that over time. We can speed this process up, though, in two different ways. Number one way is if we expose the hydrogen peroxide to light, it will break down faster, like within hours instead of days. And that's why they put hydrogen peroxide in a brown container, because that way the light can't get in and break it down faster. This does have an expiration date on it, and if you wait too long after the expiration date to use it, you'll just have a bottle of water. Just letting it sit around, it will just turn into water. It'll just break down into water and oxygen. The other thing that can speed up this chemical reaction, though, is the protein catalase. And catalase will make this breakdown happen instantly. And that's what we're going to be doing today. We're going to be using catalase to break down hydrogen peroxide into water and oxygen. By the way, the oxygen is released in bubbles. And so the bubbles is what we're going to use to judge how fast this chemical reaction is happening today. So we're going to test a few things. We are going to test some cooked liver for catalase. We are going to test some raw liver for catalase. We're going to test carrot cells, and chicken muscle cells. And my potatoes are looking a little sad, but we're going to test some potatoes as well today for catalase. All right, so two kinds of liver. Raw chicken, carrots, and potato, we're going to see how much catalase we think is in these cells. So the more catalase there is in cells, the more of the catalase or the hydrogen peroxide breakdown reaction will happen, and the more bubbles we will see. So the faster, the more catalase you have, the more hydrogen peroxide you can break down, the more bubbles you will see. So what we're going to do is we're going to see how does the type of cell, for all these different cells, how does the type of cell affect how much bubbles will form in our test tubes. So we're going to be using test tubes today, putting things in there and seeing how much bubbles will form. This is a quick review of last year, parts of an experiment. We're going to have a whole other chapter on parts of experiments in the spring, so hopefully you kind of remember this, and we'll review even more later. But the manipulated variable we're using here is the type of cells. We're testing different cells to see if it makes a difference. Our results variable is our amount of bubbles that we're seeing produced in the test tubes. Hypothesis choices. Either the cells will have different amounts of catalase, and you can maybe guess which thing you think might have the most if you think that's what's going to happen. Hypothesis or outcome 2 is all our materials might have the same amount of catalase. Hypothesis 3, none of the materials have catalase. And hypothesis 4, we've made a mistake and we shouldn't trust our results at all. That hasn't happened yet today, but who knows? Constant variables are variables in an experiment that we must keep the same. And we need those because otherwise we have more than one thing that's different and we don't know what caused our results. So in a well-designed experiment where we can trust our results at the end, we should only have one thing different. So what kids could think of today that we needed to keep the same, we're going to use the same size chunks. I tried very hard to get these chunks cut to the same size. Same person doing the experiment, same temperature in the room, same test tubes. These are all the same type and size. Same amount of hydrogen peroxide. We're going to be using about 10 milliliters of hydrogen peroxide per test tube today. All right, so I'm going to go ahead and start the procedure here. Turn my camera kind of down so you have a good view of the test tubes and show you what's going to happen here. So first of all, I need to put a chunk of each one of these things in each test tube. So this is pretty straightforward, kind of quick here. We'll put a chunk of liver into one test tube. We'll put a chunk of, I'm going to put this one on the end, we'll put a chunk of raw liver in the other test tube. Kind of takes a minute for that to slide all the way down. Better wipe off my tweezers in between here. Oh, people are coming back from lunch already. I'm still recording my video, Maggie. Piece of chicken, piece of carrot, and piece of potato. All right, I'm gonna help these two things down to the bottom of the test tubes a little bit so you can kind of see. So this is pretty straightforward. So you have these 5 different things that we're going to test. We got, let's do this order. We got potato, we got carrot, chicken, raw liver, and cooked liver. All right. So then what I'm going to do is, so there's catalase in the cells of these different living things. So what we're going to do is take our hydrogen peroxide. You can't see that there. And I'm going to measure out 10 milliliters of it and put it into each test tube. So here's 10 milliliters on our chunk of potato. And hopefully you can kind of see that. We got some fizzing. And actually a fair amount of catalase. I don't know if you can see that close up. There's little bubbles forming on the surface of the potato. It's kind of fun to watch. At home, people, if you have potato and hydrogen peroxide, go ahead and cut a little chunk up, put it in a little container, and pour some hydrogen peroxide on it, and you can kind of see how it reacts. All right, the next one we got is carrot. It's kind of funny, the potato is still reacting. I still see fizzing occurring in that test tube. Then we have the carrots, and so if I compare these two, I can tell you right away that I think the potato has more going on than the carrot. So I think a potato, look, it makes the potato go up and down, by the way. The bubbles attach to the potato piece and raise it up, and then it sinks, and then it, like the bubbles, watch, it'll sink. Then the bubbles attach to it and make it go back up again. Kind of interesting. All right. Then we've got the raw chicken. Raw chicken. Ooh, the kids are coming back from lunch, you guys, but that's okay. All right. And then we got the raw chicken. Let's see what you think. Does raw chicken have more or does potato have more? This is the potato. This is the raw chicken. The potato has more than the chicken. We can let, oh, same amount of time we should have put as a constant variable too. We'll check these back in a minute or two here. Then we have the raw liver. Let's put the 10 milliliters of hydrogen peroxide on the raw, or excuse me, this is the cooked liver. Cooked liver has not much going on at all. Look at that. All right, and then our last one. Better get this done before everybody comes in here. Our last one is the raw liver. Are you guys ready for this? I'm gonna make sure you get a good view of this. There's our raw liver, adding the hydrogen peroxide. Oh no, oh no, oh no. All right. Raw liver, by far, of course, has the most catalase. By the way, our potato is still going here. Potato is still fizzing. This one's still going. If you let this go, I'm trying to get in front of the camera there. Will it focus? You can kind of see the bubbles forming. Kind of interesting to look at it in the test tube. And the bubbles keep kind of forming on the surface of the liver. And it'll just go and go and go. By the way, the test tube gets warm. This is what we call an exothermic reaction. This gives off energy, so it will actually also make the test tube warm. All right. So if I was going to rank these, raw liver, number 1. I'm going to say potatoes, number 2. I think raw chicken is number 3. I think the carrot, got to judge between the carrot and the cooked liver. They're probably a tie for last place. Neither one of them, maybe the cooked liver has a little bit more going on than the carrot. I'd say cooked liver is 4 and carrot is 5. Let's put these in order again. So I say raw liver, potato, chicken, cooked liver, carrot, in order from 1 to 5. Kind of see that, see all the results. That raw liver is still going. Isn't that funny? All right. So when you're done with this, make sure that you use the slideshow and everything. And I think in this video, you should have heard the answers to all of the lab questions that are posted in Schoology. So you need to go to Schoology, open up the Catalase Lab questions, and then use what you learned here in the video to answer your questions. And have a great day, and we'll see you soon. Thanks for watching the video. Bye.